First of all, I'd like to thank Venture Center, TED, all the organizers of TechX, and all of you present here today for this opportunity for us to present our work. We're a startup based out of Bangalore. We are CrisprBits, and today the title of my talk is PathCrisp, which is a CRISPR-based diagnostic platform that we've made to address point of need concerns for diagnostics pan applications. And today I'll be talking to you about veterinary health applications that we have looked into. Next slide, please. So the problems addressed, of course, as mentioned, throughout the discussion before this, the lack of accessible and rapid diagnostic tools at the point of need in veterinary sector is a critical challenge, not just in India, of course, but also at global health of animals, as well as food security and public health. Livestock diseases are alone resulting in about 300 billion economic losses, which are reducing productivity, causing high mortality rates, and of course, leading to more trade restrictions. So although, I'll be talking to you about our platform, I'll also be talking to you about two use cases that we've used our platform for, which are relevant to today's context. One being salmonella, which is salmonella in meat and animal feed. And in India, that itself is a 60 million USD loss concerning matter right now. And foot and mouth disease virus, or FMDV, as it's commonly known as, again, it leads to about 0.5 to 1 billion USD loss for the farmers, as well as reduced milk productions, death of high-yielding cows and young ones, young livestock, etc. as well. Next slide, please. So I'll spend a minute or two just to try to explain to you our platform. It's we're using CRISPR, which you might have heard mostly in editing situations, but we have developed this for diagnostics. And the reason being that CRISPR comes with unprecedented precision and specificity like no other technology before. And essentially, our platform uses CRISPR-Cas12 specificity with a guide RNA to target a region. So in this case, the DNA or the RNA of the virus or the pathogen or the bacteria or any other target that you may give us in the platform. And once the Cas recognizes the region, the specific region of the target, it undergoes something called transcleavage activity, which gets activated, which cleaves any single-stranded DNA in the neighborhood. So what that means for our assay, as you can see in the slide, is that once activated because of the presence of the target, the CRISPR-Cas complex will cleave any single-stranded DNA, which we have labeled with fluorophores. This will release the fluorescence, and we can use the fluorescence as a readout for an assay. That being said, we are also building this at the same time for a lateral flow readout to really make this assay a point of need compatible assay for field testing and easy use. So if you just look at the right side of our slide, post the guide design engine, which is proprietary, which we take some time to look at the target and design very specific guides depending on the pathogen, or in this case, we've also used this for human cells. Then after sample extraction, in one particular tube, we undergo amplification, which is isothermal amplification. Currently, what I'm talking to you about is we're using LAMP. We are building this for other amplification methods as well. And then in the same tube, at the secondary level of testing, our CRISPR assay detects the same target. So there is two levels of checks and two levels of specificity that bring about the precision of the diagnosis. The readout is through a simple, benchtop fluorimeter. In this case, we've shown you a benchtop qPCR. We have tested about six different type of fluorescence readers, including different qPCR machines and handheld fluorimeters and isothermal LAMP units for this assay, and it works perfectly in all of them. And like I said, we are building this for lateral flow strips as well. So the other advantage of our assay is that we have made it lyophilized, so all in a single tube. So this reduces the whole need for cold chain transport for any of these diagnostic kits. And these are stable for long term and very easy to use, that everything comes compacted in one single tube that you need post sample extraction. We're also reducing all of the complications of sample extraction, where we are now building this with very simple on-spot crude extractions, such as FDA cards. Next slide, please. So just to repeat, it's a high specificity and sensitivity. There is rapid turnaround time. There is no thermal cycling. So it's a single temperature for as low as 60 minutes. It's a precision which is unprecedented. So the target is precisely is precisely targeted in the mix and milieu of sometimes cow DNA plus the pathogen that is present. It's versatile, can be adapted. It's a platform that we developed that can be adapted to various pathogens. We are currently trying to spatially multiplex this for many, many different targets which are similarly associated, such as AMR. And we're building it to be very affordable without the need of skilled hands, and thus allowing for more testing, more effective and timely diagnosis, and usable readouts that can affect both reduction in economic losses as well as advise clinicians and veterinarians of how to proceed with that knowledge. Next slide, please. So the use case one is to test salmonella, which in the food industry would be prevalent and a major cause for meat and animal feed going bad. So in this case, the current guidelines by FSSAI is random sampling of meat cuts, ground meat, other preparations, and sample sizes of up to 25 grams. In our assay, we are trying to build this to actually serve the need of constant testing in the facilities as well as in the feed, before it actually reaches and prevents any loss which has already reached the animal as a feed. That would be every batch on site. Next slide, please. So this is the actual data. So this you can see that the red is showing you that from Salmonella genomic DNA versus our no template control, which is our negative control, the blue, our readout is about 8 to 10-fold higher fluorescence level compared to background. Also, if you see on the right, that's a lateral flow where we can see when you see 1,000 copies of Salmonella, you can see that clearly the top band is there when Salmonella is present, whereas the bottom band is only there where it's detection negative. So this is different from a pregnancy test where you expect both the lines, one test is control. We have developed this as a molecular lateral flow readout where only a top band means positive and a bottom is negative. And when we spiked in 1 copy of Salmonella in milk, we can actually detect it, which is very promising. And we are very excited for this because, we can detect very low copies as well, really, serving the need of diagnosis to be able to reach a point of early detection and have impact in advising further. Next slide, please. So we have an ICMR grant for this, for not just Salmonella, but also all the antibiotic resistance markers that are linked with Salmonella, such as CTXM and TEM. And here is data showing you that, again, our assay built specifically for CTXM and TEM is showing you a high 8 to 10-fold fluorescence readout compared to our background, no template control. And this is working again as the platform is taking forward. And we plan to build this as a spatial multiplex panel, as I mentioned earlier. Next slide, please. So again, the second use case being foot and mouth disease virus. Here we have a principal secretary advisors grant in collaboration with Art Park, where we are developing, again, a CRISPR-based assay for detection of FMDV virus for early detection of all positive animals. So again, as I mentioned, this does lead to economic losses and to farmers, as well as reduced milk production and decreased livestock weight. So the idea is that the vet could probably carry it. The cows are insured, so this could be a regular testing. So we have, hope that this can be used regularly in combating the quick spread and also lead to less economic losses if caught early and used correctly for isolating cows and preventing further spreads. Next slide, please. So data for this, again, here we started off by building this by taking the actual vaccine sample, as well as the antigens that are used to create these vaccine samples from our collaborators. Again, this was in a collaboration with Art Park, and they helped procure these different antigens from Indian Immunologics. And as you can see, our assay is showing an immense positivity compared to background, where, again, it is very, very high fold compared to background. And when we do it with, a positive control where we topoclone and make synthetic RNA as well, we do see this. And now we are taking this forward to actually doing environmental surveillance samples, as well as, doing field testing and samples that are coming in with our collaborators with inactivated viruses in controlled biosafety level conditions. Next slide, please. So this is, again, just an example showing you that we have been sampling soil samples, wastewater samples from the farms that we are working with. And as you can see, our positive control that we're using is the vaccine. Currently, our data is showing no FMDV detected in any of these samples, and we are continuing to do this as a surveillance project. Next slide, please. So as mentioned, our current version takes the lyophilized tubes together in a portable heat block and fluorescence reader such as this. We were, this is one example called the RevoSketch, We are also, looking at we are currently working with various consultants to make composite cartridges for multiplexing, currently spatial multiplexing, and we're thinking of all sorts of different cartridges that might be applicable in microfluidics to combine a bunch of these pathogens together. And a platform like this is where we are hoping to meet, and our ask is one of those to help us build these sort of spatial multiplex platforms so that can be more useful for multiple targets in one go. Next slide, please. So again, market analytics, as you can see, there's livestock producers. This would be applicable for QC on site. This would be applicable for animal feed producers, as well as at veterinary clinics and hospitals for rapid disease detection, faster diagnosis, as well as, sort of enhanced decision making and streamlined quality assurance, as I mentioned. Next slide, please. So this is just a list of our patents. We have about 5 patents, and we also have 2 papers. We are currently also submitting some of our work in other journals. The second paper is right now in review under scientific reports, and we continue to build this as a platform for various other pathogens as we speak. Next slide, please. So this is our team. And as you can see, it was initially founded by 5 distinguished alumni in BITS Pilani. And we currently have PhDs and master's students from molecular biology from across St. John's, NIMHANS, TIGS, NCBS, INSTEM. Dr. Vaijayanti Gupta is the COO. She comes from a lot of experience from Strand Life Sciences. I head the R&D, and Vandana, who's also joined me in this call, heads our business development. She also comes with a lot of experience from Bristol Myers Squibb, and also in India, she has led product development largely across different companies. Our team comes from an experience across different molecular biology sectors. So, it's a great bunch of scientists, and we are taking diagnostics as well as gene therapy forward with a bunch of scientific advisory board members who come from the CRISPR space, as well as infectious disease space, as well as gene therapy space. Next slide, please. And so this is our founders, as I mentioned, including Dr. Chandru, who, led Strand Life Sciences for about 15 to 20 years. And then finally, now this is his second company that he has started, along with his friends from BITS Pilani. And all of them are distinguished alumni, currently leading, successfully have led other companies as well. And the scientific advisory board, as I mentioned, includes Professor Sam Sternberg, who was a PhD student in Jennifer Doudna's lab herself and now runs his own lab at Columbia in New York. Dr. Nancy Cardori is infectious disease specialist at Virginia Norfolk Hospital, and she guides us on the infectious disease panel. And Dr. Roger Haja, Ajit Chande, and Dhananjay help us with the gene editing space. Next slide, please. So basically, next steps is that we have developed this platform technology. We've developed and demonstrated proof of concept at lab scale and currently in validation stage. So our next phase would include, of course, we have identified manufacturing partners, and we are in the next stage of, signing with them and taking it for beta testing, and actually then getting all the regulatory approvals and commercializing and launching it for, some of the use cases I mentioned and some others as well. We have also, I would like to mention here, we have developed a test for sickle cell anemia. This has been challenging because in that case, you have to identify a single nucleotide polymorphism, which is very interesting because we have used CRISPR's precision to be able to detect one single base change. And this has been very exciting because going forward, a lot of these pathogens mutate at just a single point and the diagnostic assay becomes defunct. So that is something we know that we can quickly adapt to. And also, for antibiotic resistance, that's very valuable. So our ask for today, of course, is that for industrial partners interested in sponsoring further technology development and scale up, it would be great, specifically the microfluidics and multiplex engineering multiple format. Of course, funded project support focusing on a QC method for food exporters or traders, a funded project aimed at developing a QC method for incoming animal feed and feed additives, and lastly, any partners interested in paid pilot programs leading to purchase order. Next slide, please. Yes, for more information, you can contact us through Pradnya or directly reach out to CrisprBits at info@CrisprBits.com. Thank you. Happy to discuss any questions.